Hello guys, welcome to Dr. Science. So today we are going to talk about toll-like receptors. So first I will talk about basics, what are meant by toll-like receptors. So here is a neutrophil. So here is a neutrophil and this neutrophil is having certain types of receptors present on it. Okay. So here is a neutrophil and as you can see here is a bacteria, and this bacteria is having certain types of pattern. Okay, like here is a green pattern, here is purple pattern, and here is your red pattern. This pattern is nothing but your antigens which are present on microbes. So in order to recognize this pattern, the neutrophil is having the pattern recognition receptors. So this pattern recognition receptors, one such type is your toll-like receptors. Okay, I talked about pattern recognition receptor in my previous video, so please watch that video in order to understand what is meant by pattern recognition receptor, what is meant by pathogen associated molecular protein, and what is meant by damage associated molecular patterns. Okay, so as you can see here, neutrophil is having certain types of pattern recognition receptors which will help to identify the pattern present on microbes, and one such type of pattern recognition receptors are your toll-like receptors. And not only that, toll-like receptors were first found in Drosophila. Okay, Drosophila is a fly, and first they found these toll-like receptors in Drosophila, and they found nearly 10 types of toll-like receptors. So what is the function of these toll-like receptors? We will see in a second. So basically, suppose this is the plasma membrane. The plasma membrane is going to have certain type of toll-like receptors, and not only that, in the cytosol also there are toll-like receptors, and on the top of that, suppose this is the endosomal vesicle. This endosomal vesicle is also having toll-like receptors present inside it. So why there are three types of toll-like receptors present in different location? The answer is simple. Suppose this is a toll-like receptor which is present on the plasma membrane, and suppose there is a microbe here, there is a bacteria here. Obviously, then the toll-like receptors which are present on the plasma membrane, they will get activated and they will kill this bacteria. So basically, toll-like receptors which are present on the plasma membrane, they help to kill extracellular microbes. Okay, whereas when the bacteria enters into the cytosol or inside the cell, the bacteria can either get killed by the cytosolic toll-like receptors, or the bacteria can enter into the endosomal vesicle, and there it can be killed by the toll-like receptors present on the endosomal vesicle. Okay, so we will see how the toll-like receptors are working. So suppose this is the toll-like receptor, this is the plasma membrane, and on the plasma membrane there is a toll-like receptors, and this toll-like receptor is going to get activated by certain type of bacteria. So once the toll-like receptor is activated, it will go and activate two types of transcription factor. So what is meant by a transcription factor? I will just give you a brief lecture on it. I will not go deep into the biochemistry part. So here is a DNA, and this DNA is going to have a lock here. So here there is a lock, And obviously if there is a lock here, the DNA cannot synthesize anything. So the DNA cannot get converted into mRNA, which cannot get converted into mRNA, and this process which is in which DNA is getting converted into mRNA, it is called as transcription. And from mRNA it cannot synthesize proteins such as your cytokines or your adhesion molecules. I will talk about them, just wait a minute. Adhesion molecules, and the process of converting mRNA into proteins is called as the translation. So as you can see here, there is a lock, hence the DNA is not getting converted into mRNA, and from mRNA proteins are not getting formed. So we need a key, right? We need a key to unlock the DNA synthesis, and this key is called as your transcription factors. So once the transcription factors goes and unlock this key, obviously the DNA synthesis will occur, and from DNA synthesis we will form mRNA, and from mRNA we are going to form proteins such as cytokines and the adhesion molecules. And such type of transcription factors are the two types of transcription factors once the toll-like receptors are activated are your Nfkp pathway, and not only that, the interferon regulatory factor 1 or IRF1. So once the Nfkp pathway is going to get activated, so the Nfkp transcription factor have been activated and it will produce proteins, right? And one such proteins are it will increase cytokines and it will increase the adhesion molecules. So what are cytokines? So I will talk in basic terms. Suppose here there is a guy, and this guy is having a deodorant or perfume. When the guy is having a deodorant or a good perfume, obviously the molecules are going to get diffuse around his surroundings, and eventually the girls will get attracted to him, right? The functions of cytokines is also similar to this. Okay, suppose here is a microbe which is having a lot of antigens, and this microbe is going to get activated, is going to activate the toll-like receptors present on the neutrophil, and once the neutrophil is going to get activated, it will activate the Nfkp pathway and will produce some cytokines. And once these cytokines are released, the macrophages, which are effective in killing, and not only that, the T lymphocytes and the macrophages, which are effective in killing bacteria, they will get attracted to these cytokines, and so once they get attracted to these cytokines, they will surround this bacteria, and the neutrophils, the bacteria, and all these will kill the bacteria. The neutrophils, the T lymphocyte, and the macrophages, they will kill this bacteria. So cytokines are like they are like scent molecules, okay, which will attract, like I already said the theory about deodorant. They will attract these T lymphocytes or macrophages to the site of infection. We will be talking about cytokines like interleukin 1, interleukin 6, interleukin 4, 5 and 13, etc. in the coming videos. So you have to trust me on that one. So not only that, I told you one other function which is to increase the adhesion molecules. So what are these adhesion molecules? In order to understand this one, I will draw a blood vessel. So this is a blood vessel, as you can see here, this is the blood vessel, and this blood vessel is lined by endothelium, right? The blood vessels are lined by endothelium. So what happens when a neutrophil is going through this? Normally the endothelium, it is non-sticky. Okay, by non-sticky I means the neutrophil is not going to stick to this endothelium because it is not having the adhesion molecule or the sticky molecules. But I already told you, once the Nfkp pathway is activated, it will increase the sticky molecules, which are called as the adhesion molecules. So once the sticky molecules are expressed on the endothelium like this, the neutrophil will go and get attached to the sticky molecules or the adhesion molecules, and once they are, let me clear this one. So once they are going to stick to the endothelium, obviously the neutrophils can diffuse and go towards the site of infection. So here is the bacteria. So the neutrophils are going to stick to the endothelium, and once they stick to the endothelium, they can travel to the site of infection. Now these sticky molecules or the adhesion molecules are nothing but your selectins or integrins. Okay, so what is the function of Nfkp pathway? Once they are increasing the adhesion molecules like selectins and integrins, and not only that, they are increasing the cytokines. So I will draw a basic diagram here. So this is the blood vessel. So what is happening here is the site of infection, and the neutrophils, they have to stick to the endothelium. Once they are going to get stick to the endothelium, they will be running towards the site of infection. So once they are towards the site of infection, they will release certain types of cytokines. Once they release the cytokines, they are going to attract the macrophages and the T lymphocytes. Okay, so this is the toll-like receptors. I talked about two transcription factors. One was the Nfkp pathway. The other was the interferon regulatory factor 1, interferon regulatory factor 1, or the IRF1. Now the function of the interferon regulatory factor 1 is to synthesize your synthesize the interferon type 1 interferons. The functions of interferon regulatory 1 is to synthesize the type 1 interferon. So what is the function of this type 1 interferon? So as you can see here is a guy. This guy name is type 1 interferon. So the type 1 interferon, the function of the type 1 interferon is to convert the viruses, to convert the active virus into inactive virus. Hence type 1 interferons, they are also called as your antiviral interferon. Okay, so once again I will give you a summary of what is happening in case you spaced out. So this is toll-like receptor. So toll-like receptors are activated. Once they are activated, they are going to activate the Nfkp transcription factor, and not only that, the interferon regulatory factor 1. So the Nfkp pathway is activated, they will increase the cytokines, and not only that, they will increase the adhesion molecules. The interferon regulatory factor 1 will increase the synthesis of type 1 interferons, which are your type 1 interferons, which are also called as antiviral interferon. Okay, so let's watch this diagram here. So as you can see here, there are different types of toll-like receptors present on the plasma membrane, toll-like receptors present on the cytosol, toll-like receptor present in the endosome. Okay, now you can see here there are lectin pathway, the C-type lectin pathway. I will talk about the C-type lectin pathway, and not only that, the nod-like receptor in my next video. So thank you guys and have a great day.